So why does the urea cycle take place in the first place? The human body generates a lot of waste products, and our kidneys are capable of getting rid of most of them. However, the kidney can't deal with ammonia on its own, but the liver comes to rescue. This ammonia is toxic to the body and it cannot be excreted properly by the body. So the urea cycle is a biochemical pathway in the body which converts the free ammonia into urea, which is comparatively non-toxic to ammonia. It is then transported to the kidneys and can be easily excreted by the body through urine. So where does the urea cycle take place? It occurs mostly in our hepatocytes in our liver cells, but also in smaller extent in our kidneys. Ammonia is sent through the urea cycle in the liver, which is a series of enzymatic reactions that convert it into urea. Urea then can be easily dealt by the kidney. The urea cycle has 5 steps. The first step being the formation of carbonyl phosphate. This occurs in the matrix of the mitochondria. The amino acid arginine will be processed by the enzyme arginase. Arginase is unique to the liver and is a very important enzyme that is necessary for the urea cycle to occur. Arginine gets acted on by arginase to form orthonine, another amino acid. In doing so, it removes part of the backbone of arginine that includes 2 amino groups, and that's what actually produces urea. The ammonia then gets brought into the mitochondria along with CO2 that comes from the bicarbonate. CO2 and the ammonia will then be acted on by an enzyme known as carbonyl phosphate synthetase 1 to form carbonyl phosphate. Then the carbonyl phosphate will be added to the orthonine by an enzyme known as orthonine transcarbamylase. The second step is synthesis of citrulline. This also occurs in the matrix of the mitochondria. OTC take orthonine and carbonyl phosphate and add them to the citrulline. Citrulline is easily permeable to the mitochondrial membrane and therefore it diffuses into the cytosol. The third step is synthesis of argininosuccinate, and this takes place in the cytoplasm. Citrulline will be acted on by the enzyme argininosuccinate synthetase. This enzyme needs aspartate as well. What it will actually do is produce argininosuccinate. Argininosuccinate will then be acted on by the enzyme argininosuccinase to produce arginine.
The fourth step is the cleavage of arginosuccinate. There are some carbons missing between arginosuccinate and arginine. The reason for this is another product of the arginosuccinase reaction called fumate. So a part of the carbon skeleton of arginosuccinate is actually recycled and it becomes fumate. Now furate can be used for the citric acid cycle, or it can be converted to malate and oxaloacetate, which can either be used for glucogenesis or it can be used to produce more aspartate via the enzyme aspartate aminotransferase. So as you guys can see, the cycle can continue if you keep having aspartate. And in fact, you need a high amount of aspartate for the urea cycle to operate. The fifth step is the cleavage of arginine. In the final step where urea is formed, arginine is broken down by the enzyme arginase and hydrolyzed by a water molecule. Urea is removed and ornithine is also formed. Ornithine is recycled back into the matrix of the mitochondria for the next cycle. Then carbonyl phosphate synthease happens. Because CPS1 is an irreversible enzyme, it must be regulated. This is very important to the proper function of the urea cycle. What regulates CPS1 is something called an acetylglucamate, which is an activator of CPS1. This comes from the acetyl-CoA and glucamate being added together by the enzyme N-acetylglucamate synthase. Arginine and starvations are activated of N-acetylglucamate synthase. Starvation is an activator because your body is starting to utilize more proteins as an energy source. That means your body needs to excrete more nitrogenous waste. Arginine and starvations are activators of N-acetylglutamine synthase. Starvation is an activator because your body is starting to utilize more proteins as an energy source. That means your body needs to excrete more nitrogenous waste. So, how much ATP does the urea cycle cost? The first place where we see the cycle utilize ATP is at the carboxyl phosphate synthase reaction. CPS1 enzyme requires 2 ATP, and because of this it becomes an irreversible reaction. Once the hepatide produces carboxyl phosphate, it cannot go back to ammonia and CO2. The other enzyme that requires energy is the aginosuccinate synthase reaction, which is the third step in the cycle, which requires 1 ATP, but in actuality it's equivalent to 2 ATP because you're actually converting ATP into MP. The ultimate cost of making a molecule of urea is 4 ATPs. The overall equation of urea formation is NaH4 plus CO2 plus aspartate plus 3 ATP, which gives you urea plus fumarate plus 2 ADP plus 2P plus AMP plus PPE. The urea cycle brings 2 amino groups and HCO3 together to form urea. Therefore, toxic insoluble ammonia is converted into non-toxic water-soluble extractable urea. Hence, a urea cycle disposes 2 waste products, NH4 and HCO3. This fact suggests that the urea cycle participates in the regulation of blood pH, which depends on the HCO3/H2CO3.